Welcome to this crash course on General Biology 2. Let's get started. There are three domains of life: bacteria, archaea, and eukaryotes. Bacteria and archaea make up the prokaryotes, which vastly outnumber eukaryotes. The key features of prokaryotes are that they are unicellular and similar in size, divide by binary fission, and have no nucleus and few or no membrane-bound organelles. Eukaryotes tend to be multicellular and more diverse, divide by mitosis and meiosis, and have a nucleus and membrane-bound organelles. Archaea are considered more closely related to eukaryotes based on similarities in their gene sequences. Bacteria are classified by their metabolic pathways, shape, growth environment, gene sequence, and cell wall structure. Nearly all bacteria contain a cross-linked polymer called peptidoglycan, not found in eukaryotes or archaea. If they have a single cell membrane and a single thick layer of peptidoglycan surrounding the cell, they're considered gram-positive and turn bluish-purple when stained with dye. An example is Staphylococcus aureus, which is abundant on the skin and can cause wound infections. If surrounding the cell is a peptidoglycan layer in between two cell membranes, the bacteria is considered gram-negative and turns reddish-pink when stained with dye. A couple examples are cyanobacteria, which generates oxygen necessary for life, and E. coli, which are mostly harmless but can sometimes cause serious diseases and food poisoning. Some antibiotics act to fight bacterial infections by inhibiting peptidoglycan polymer synthesis, and some cells in the immune system also recognize peptidoglycans. There's also a process called lateral gene transfer, where prokaryotes exchange genetic material with each other. This means that some prokaryotes can appear to be more related based on a single gene they share, even if they're actually distantly related. Thus, we can more accurately compare the relatedness of prokaryotes using stable core genes, which are genes that are unlikely to be transferred between prokaryotes and will only pass down when prokaryotes replicate. Additionally, prokaryotes can form communities called colonies. An example is bacterial biofilms, where prokaryotes bind to a surface and begin to replicate and secrete a sticky polysaccharide matrix, or biofilm, which holds them all together in a large group. These can form on teeth as plaques, artificial joints, contact lenses, and more. Going back to archaea tend to be distinguished by a unique lipid structure contained in their cell membranes, which have ether linkages instead of ester linkages seen in bacteria and eukaryotes. Their cell membranes are also monolayers, so only one layer of lipids, unlike the phospholipid bilayer consisting of two rows of opposing phospholipids seen in bacteria and eukaryotes. There are also viruses, which have still not been definitively classified as living or dead since they cannot replicate without a host, but they share lots of resemblances with organisms we call living. They typically consist of DNA or RNA surrounded by an envelope protein layer containing surface proteins that allow them to bind to and infect prokaryotic and eukaryotic cells and replicate themselves using the machinery of their infected host cells. Returning to eukaryotes, they are very structurally and functionally diverse and consist of plants, animals, fungi, and protists. Protists are defined as any eukaryote that is not a plant, animal, or fungi. The main features of eukaryotes are increased cellular compartmentalization, a more flexible cell surface, a complex cytoskeleton giving eukaryotic cells their shape, a nuclear envelope, and digestive vacuoles which can ingest nutrients, water, and waste. Most notably, though, is how eukaryotes gain their internal organelles, which is explained by the endosymbiotic theory. The endosymbiotic theory states that at some point, an ancestral eukaryote engulfed and retained a bacteria, which turned into key parts of the cell over time, the organelles called mitochondria and plastids. These proved to be a key evolutionary advantage for eukaryotes, since they provided the cells with energy. In general, eukaryotic cells that directly engulf the bacteria are said to have undergone primary endosymbiosis, whereas eukaryotic cells that indirectly engulf these bacteria by engulfing a eukaryote already containing an engulfed bacteria are said to have undergone secondary endosymbiosis. These engulfments occur via phagocytosis, which is where a part of the plasma membrane of the cell engulfs the bacteria and then pinches off, forming a vacuole or phagosome with a membrane derived from the original plasma membrane. Going back to protists, protists are enormously diverse, with most being unicellular and microscopic, and some being multicellular and quite large. These include plankton, or free-floating aquatic organisms, phytoplankton, or photosynthetic plankton, and autotrophs and heterotrophs. Autotrophs get carbon atoms and energy from inorganic matter, such as CO2, whereas heterotrophs get their carbon atoms and energy from organic compounds made by other organisms. There are also diatoms and dinoflagellates. Diatoms are photosynthetic producers found in coastal and fresh waters, and their buried remains are used as fossil fuels. They also have amazingly intricate appearances due to silicon deposits in their cell walls. Dinoflagellates are unicellular and major photosynthesizers, mostly found in oceans and fresh water. As a major part of the Earth's phytoplankton, they produce food for many aquatic species and much of the oxygen in the atmosphere. They also have a complex cytoskeleton and two tail-like structures called flagella, hence the name dinoflagellate, where di stands for two and flagellate corresponds to flagella. They also sometimes undergo massive replication and release pigment molecules, which can cause red tides that look really cool but are toxic to other organisms in the water. Now, let's talk about fungi. One synapomorphy, or trait unique to fungi, is absorptive heterotrophy, where fungi secrete enzymes into the environment to break down nutrients and then absorb these nutrients. Another synapomorphy is the presence of chitin in fungal cell walls, which is a polysaccharide polymer which adds structure and rigidity to fungal cells. Fungi can be multicellular, like mushrooms, or unicellular, like yeast. Mushrooms are spore-producing, or fruiting, structures that have a vast underground network of branches called mycelia. These mycelia consist of filaments called hyphae, which absorb nutrients. Fungi can also be defined as subropic, parasitic, pathogenic, and mutualistic. Subropic fungi, sometimes called molds, are major decomposers and key to Earth's carbon recycling, and get their nutrients from non-living organic matter like dead plants. Parasitic fungi invade plants and steal nutrients from them by absorbing them through their hyphae. Pathogenic fungi can cause diseases like fungal infections, and mutualists benefit both the fungi and plant since they help the plant absorb water and nutrients while also getting some of the food made by the plant. Additionally, fungi reproduce both sexually and asexually, going through a complex life cycle consisting of haploid or N, diploid or 2N, and dikaryotic or N plus N stages. The concept of haploid and diploid was discussed in our General Biology 1 video, and dikaryotic basically means that the fungal cells contain two nuclei, but they're not fused like diploid cells are. Moving on to animals, what makes animals so special? Well, animals are incredibly diverse and have the characteristics of being multicellular heterotrophs, and they have internal digestion and typically the ability to move. Animals are thought to originate from choanoflagellates, which share similarities with sponges, which are simple animals. When we distinguish animals, we can look at several features, including body symmetry and germ layers. Looking at body symmetry, animals can be asymmetrical with no symmetry, radially symmetrical with symmetry in every plane, and bilaterally symmetrical with symmetry only in one plane, like left and right. Looking at germ layers, these are the layer of cells that form in animal embryos and eventually give rise to an adult organism. Diploblasts have 2 germ layers, while triploblasts have 3. Triploblasts can be further categorized into protostomes and deuterostomes, where protostomes form their mouth first, and deuterostomes form their mouth later in development. Protostomes are the most diverse group of animals that include insects, and deuterostomes are made up of 3 main groups called echinoderms, hemichordates, and chordates. Looking at chordates, these include vertebrates, which have a backbone called the vertebral column. Within vertebrates are amniotes, which include species like reptiles, birds, and mammals that lay so-called amniotic eggs surrounded by a protective membrane. The key features of mammals are sweat glands, mammary glands, hair, and a 4-chambered heart that fully separates oxygenated blood from deoxygenated blood. Let's talk about plants now. Plants are multicellular autotrophs that contain cell walls, mitochondria, and plastids. Plants can be divided into algae and green plants, where green plants include land plants. Land plants can be classified as non-vascular or vascular, where vascular plants have internal tissues that allow them to move water and nutrients through the plant. An example of non-vascular plants are mosses. Inside vascular plants are non-seed plants and seed plants. Non-seed plants, like pine trees, are called gymnosperms, meaning they don't produce flowers. And seed plants, like roses and orchids, are called angiosperms, since they produce flowers. The key adaptations of land plants are protected embryo, which prevents drying out, a waxy coating called a cuticle on their leaves, which allows them to retain water, and closable openings called stomata, which allow them to exchange gases. They can also form associations with fungi to better absorb water and nutrients. Like we discussed with fungi earlier, plants also have a life cycle, this time only consisting of a haploid end cycle, where cells make up the so-called gametophyte, and a diploid 2-end cycle, where cells make up the so-called sporophyte. The gametophyte is where photosynthesis occurs and energy is created, and the sporophyte is nutritionally dependent on the gametophyte. Ferns, mosses, angiosperms, and gymnosperms all have their own unique life cycles, but they all generally have a haploid gametophyte generation and a diploid sporophyte generation. Talking more specifically about angiosperms, or flowering plants, how do plants know when the right time to flower is? Well, they have photoreceptors which respond to light at specific wavelengths. These photoreceptors contain special proteins called phytochromes, which tend to mostly absorb red light and far red light from the sun. Phytochrome cycles between an inactive form called PR, which absorbs red light, and an active form called PFR, which absorbs far red light. With prolonged dark exposure at night, PFR is converted into PR. Plants that flower only when the night is shorter than a certain threshold are called long day plants, since they like more daylight and less darkness. Now let's discuss photosynthesis, a process responsible not only for the survival of plants, but pretty much all life on Earth, including you and me. Photosynthesis, or light synthesis, is a process which takes sunlight and carbon dioxide and converts it into sugars and oxygen. The plants use the sugars as energy, and the oxygen they produce goes into the atmosphere, which we humans breathe to stay alive. Photosynthesis consists of two parallel pathways, the light reactions and the carbon fixation reactions. The light reactions occur in the thylakoid membranes inside the chloroplast of plants. Light hits the pigment molecules called chlorophylls, which act as a light harvesting system, and this sets off an electron transport chain that eventually produces the energy-filled molecule ATP and oxygen as a byproduct. The light reactions also occur in the chloroplast of plants, more specifically in the fluid called the stroma. A key enzyme called rubisco speeds up the reaction, where carbon dioxide is converted into various carbon intermediates, which are used to make sugars like glucose. Returning to animals, animals eat organic or carbon-based matter, like plants, to stay alive. They have a so-called metabolic rate, or rate of energy consumption, which determines how much they eat and when. This metabolic rate can be measured by how much oxygen they can consume. Additionally, animals that maintain stable internal conditions like body temperature are called regulators or endotherms. Endotherms include mammals and birds, and these keep their body temperature at a certain level regardless of the temperature in their external environment. Animals like frogs, lizards, and many fishes are called conformers or ectotherms, since they just let their internal temperature match the external temperature. Animals, particularly endotherms, have many mechanisms to generate and preserve heat if they're too cold or get rid of heat if they're too hot. When they're cold, they shiver to produce heat and constrict their blood cells to preserve heat. When they're hot, they pant or sweat to get rid of heat. Animals in colder environments tend to have insulated bodies with lots of fat and fur to prevent heat loss, whereas animals in hotter environments tend to have much less insulation and a greater surface area in order to get rid of heat. Additionally, animals have a system called negative feedback, which allows them to keep their body temperature and other processes close to or at a certain level, called the set point. For example, if body temperature is above the set point, the animal will try to bring it down, and if the body temperature is below the set point, it will try to bring it up. There's also positive feedback, where increasing levels stimulate even greater increases in a process. The mammalian birth process is an example, where the hormone oxytocin is released to increase uterine contractions, which then cause more oxytocin to be released, which stimulates even stronger contractions. Our bodies have two types of glands: exocrine glands, which secrete substances through ducts, and endocrine glands, which secrete hormones into the blood. An example of exocrine glands are sweat glands, and we'll discuss endocrine glands like the pituitary, thyroid, and adrenal glands. Hormones are chemical messengers secreted by endocrine cells and cause slow-acting but powerful effects after binding to receptors in target cells. Starting with the pituitary gland, it's a pea-sized gland attached to the hypothalamus in the brain, which gives the pituitary the name of master endocrine gland as it connects the nervous and endocrine systems. It's divided into two lobes, the anterior or front pituitary gland and posterior or back pituitary gland. Both release various hormones that have different effects in the body. For example, the posterior pituitary releases the hormone oxytocin, which we just discussed, and the anterior pituitary mediates processes such as our fight or flight responses elicited by our adrenal glands, metabolism controlled by our thyroid glands, and reproduction controlled by our reproductive organs. To mediate these processes, the anterior pituitary uses, respectively, the adrenal gland stimulating hormone ACTH, the thyroid stimulating hormone TSH, and the reproductive organ stimulating hormones FSH and LH. Speaking of reproduction, the pituitary gland hormones stimulate the gonads, or ovaries and testes, to release their own hormones. These include progesterone, estrogen, and testosterone, and they regulate gamete formation, or the formation of eggs and sperm in males and females. In this way, the pituitary indirectly regulates the reproductive system. It also regulates the ovarian and menstrual cycles in females. The ovarian cycle consists of one mature egg being released by the ovaries every 28 days or so in humans. The menstrual cycle consists of the strengthened lining of the uterus in preparation for the potential arrival of the developing embryo. If the egg is not fertilized, bleeding and other unpleasant symptoms occur in what is called menstruation or menses. If the egg is fertilized, the embryo is then implanted into the uterine wall and eventually grows into a baby. The embryo will also release hormones like progesterone and estrogen throughout, and it will also release a hormone called human chorionic gonadotropin, or HCG, which is what pregnancy tests test for to see if someone's pregnant. Moving on to the digestive system, our digestive system digests and absorbs food molecules to use as energy, reabsorbs water and salt, and gets rid of waste. The foregut consists of the mouth, esophagus, and stomach. The midgut consists of the small intestine. The hindgut consists of the large intestine, and the accessory organs are the pancreas, liver, and gallbladder. Food enters our mouth where we start to break down carbohydrates. It travels down our esophagus into our stomach, which secretes hydrochloric acid and enzymes to further disintegrate the food and starts digesting proteins. From there, the food enters the small intestine, which finishes protein and carbohydrate digestion. It also starts fat digestion, which is finished by the pancreas and gallbladder, and is responsible for almost all nutrient absorption. The liver also plays a key role, removing toxins from the blood that flows out of the stomach and small intestine. The leftover blood then passes through the large intestine, which reabsorbs water. Any leftover waste products pass into the bladder and rectum, where we excrete them. Additionally, when we eat food, our blood sugar levels are regulated by two hormones, glucagon and insulin, which are released by the pancreas into the blood. Glucagon is released to bring up our blood sugar when our blood sugar level falls by stimulating the release of glucose into the blood. Insulin, on the other hand, brings down our blood sugar by stimulating the uptake of glucose into cells, which then store the glucose in a different form called glycogen. We get fuel and nutrients from what our digestive system digests, but even more important is keeping a steady supply of oxygen and getting rid of carbon dioxide. Enter our breathing system, without which we die in minutes. So, how do we breathe? An in-breath is where the diaphragm, a muscle right below our lungs, expands the chest cavity, pulling air into the lungs. On the out-breath, the diaphragm relaxes, allowing the lungs to recoil, and air is pushed out. Throughout the process, the oxygen from the air in the lungs passes into the blood through little sacs in the lungs called alveoli to be used as energy, while carbon dioxide waste from the blood passes into the lungs to be breathed out. A system closely connected with the breathing system is the circulatory system, which includes the heart. Mammals like us have a four-chambered heart that allows us to separate deoxygenated blood, or blood without oxygen, from oxygenated blood, or blood containing oxygen. The basic circuit consists of the following. Oxygenated blood from the lungs travels through the pulmonary veins and enters the left side of the heart into the left atrium. The left atrioventricular valve opens, allowing blood to enter the left ventricle, and then quickly shuts. Blood from the left ventricle then moves into the aorta, which pumps the oxygenated blood into the rest of the body. The body then uses up the oxygen as energy and sends the now deoxygenated blood back through the vena cava and into the right side of the heart, where the blood enters the right atrium. The right atrioventricular valve opens, allowing blood to enter the right ventricle, and then quickly shuts. This deoxygenated blood then moves into the pulmonary artery, which transports it to the lungs to be oxygenated again, and the cycle starts over. All of this happens in a matter of milliseconds, and this cycle is called the cardiac cycle. When blood flows into the heart, this is called diastole, or relaxation, and when blood is pushed out of the heart, this is called systole, or contraction. Additionally, the opening and closing of the atrioventricular valves causes the lub-dub sound that you feel when you put your hand over your heart. Also note that veins carry blood to the heart, while arteries carry blood away from the heart. Speaking of blood, the blood is also important for the exchange of gases, fluids, and solutes, or dissolved substances. This exchange happens in the capillaries, which are tiny branching blood vessels that connect arteries and veins. Substances like glucose, oxygen, and carbon dioxide are easily exchanged with the surrounding tissues here, since capillaries are so permeable and have such a large surface area. The fluid inside the capillaries is called the vascular fluid, or blood plasma, and the fluid outside is called the extravascular fluid. There are also various pressures present, and these promote the exchange of these gases, fluids, and solutes. The pressure from the blood is called the hydrostatic pressure, and the pressure from proteins in the blood is called the oncotic pressure. Both the capillaries and tissues exert these pressures on each other for a total of 4 opposing pressures: the capillary hydrostatic, capillary oncotic, tissue hydrostatic, and tissue oncotic pressures. Depending on the total net pressure, gases, fluids, and solutes will move into or out of the capillaries. Now, let's talk about neurons and membrane potentials. Neurons are the cells of the nervous system, and they consist of 4 main parts: the dendrites, the cell body, the axon, and the axon terminal. The dendrites receive signals called axopotentials from other neurons and transmit them to the cell body. The cell body, which contains the nucleus and organelles of the neuron, receives these signals and sends them down the axon. At the axon terminal, the end of the axon, the signals then pass down to the next neuron. Neurons have a voltage which dictates when they fire a signal to another neuron, and this voltage is dictated by the difference in the charge inside and outside the membranes of neurons. The main ions responsible for the charge difference are Na+, or sodium, K+, potassium, Cl- chloride, and Ca2+, or calcium. When positive ions are flowing into one side, or negative ions are flowing out from that side is considered positively charged. If positive ions are flowing out of one side, or negative ions are flowing into that side is considered negatively charged. If you'd like a more in-depth look into neurons, membrane potentials, and the nervous system, check out our Intro to Neuro video. Now, let's move on to a subject that has become all too relevant in recent months: the immune system. The immune system is what keeps us healthy and allows us to avoid disease by destroying invading pathogens. Basically, the immune system's job is to distinguish the body's cells from invading cells and destroy the invaders with as little damage to the body cells as possible. The 2 types of immunity are innate immunity and acquired immunity. Innate immunity is much quicker than acquired immunity, since it's the immunity we naturally have at all times. This includes the skin, mucus, chemicals like cytokine, which are responsible for our inflammatory response, and phagocytes, which pretty much engulf any invaders they come in contact with. Acquired immunity is much slower, since it has to be tailored to a specific pathogen, like the SARS-CoV-2 virus that causes COVID-19. It consists of two components, the humoral response and the cell-mediated response. The humoral response consists of cells called memory B cells remembering the pathogen and then producing plasma B cells which produce antibodies specifically aimed at fighting the specific pathogen. The cell-mediated response consists of T killer cells binding to the specific pathogens and destroying them. Our immune responses to viruses like SARS-CoV-2 uses both the humoral and cell-mediated response to destroy the virus. Moving on to a completely different topic, let's talk about animal behavior. The way animals behave have two types of causes, proximate causes and ultimate causes. Proximate causes are the immediate physiological processes and mechanisms involved in behavior, in other words, how the animal carries out a behavior. On the other hand, ultimate causes are the evolutionary advantages that lead to the behavior, in other words, why the animal carries out a behavior. Animals often show fixed action patterns that cannot be learned or modified by learning and are carried out to completion. An example is a spider spinning a spider web. It makes a perfect web pattern every time without learning at all. Learning, on the other hand, is how organisms modify their behavior due to prior experiences. A classic example is classical conditioning in Pavlov's dogs. Basically, a guy named Pavlov got dogs to learn to associate a bell with food. At first, they'd only salivate in response to being presented with food, but after some training, they would start to salivate upon hearing the bell. I highly recommend looking up Dwight Altoids on YouTube for a hilarious example of classical conditioning in action. Hormones can also have a huge impact on behavior, like oxytocin, which promotes care and bonding in mammals. Other cool examples are behavioral imprinting and the waggle dance of bees. Behavioral imprinting is basically where certain animals like ducks will attach to anything it sees as a parent during a very specific period of development called the critical period. So if the duck sees a human during this critical period, they will take that human to be their parent and will follow them everywhere. The waggle dance is a really interesting behavior where bees do a specific dance that tells other bees exactly where food is in relation to the hive. Finally, let's talk about ecosystems. Ecosystems consist of all the organisms in a particular habitat and the physical environment they live in. When describing the diversity of different communities, we can consider the richness and evenness of the species. The richness is the number of different species present, and the evenness is the distribution of species, in other words, how abundant each species is. These communities can change over time due to factors like colonization, extinction, natural events, and human influence. They can also interact within themselves in chains called trophic cascades. Primary producers like grass, berries, and roots on the bottom often carry out photosynthesis. Primary consumers like rabbits and mice eat these primary producers, and above these primary consumers are omnivores, which eat both the primary consumers and primary producers. Finally, you have secondary and tertiary consumers, which continue the trend, eating the animals below them on the food chain. The amazing part about trophic cascades is that everything is so interconnected. An effect on one part of the food chain, no matter how small, can have an effect on another part of the food chain. And that's it. I hope you found this video on General Biology 2 helpful.